let's get into these whole genome duplication events. So in all of these sort of macroevolutionary patterns of gene and genome evolution, it's really important to consider that all of this is taking place within an ecological context. And so that's the idea that this diagram from Seagrave's 2017's review on whole genome duplication events is trying to convey. So the larger plant is the one that has undergone a polyploidization event, and sometimes whole genome duplication events or polyploidization events are accompanied by a change in the floral morphology or coloration, which could result in a change in which pollinators are attracted to the plant. The cells that make up the roots will sometimes change in configuration just as a result of the changing cell size. The root exudates that are being secreted into the rhizosphere might change, and there's the potential that there could be a change in the association of mycorrhizal fungi and or bacteria. Seeds will often be a different size in polyploids, so they could potentially attract different seed dispersers or seed predators. Just a whole host of really cool different changes that can accompany this. And so you're welcome to kind of browse through this table that I copied from Seagrave's. It's really, very complete. It's a really kind of holistic look at the potential changes that can accompany whole genome duplication events. Obviously the stoichiometric changes, this is the whole reason that I went on that little diversion on ecological stoichiometry theory and in the beginning of the lecture. If the genome size increases in plants, it tends to result in an increased requirement for nitrogen per unit biomass and an increase in the requirements for phosphorus per unit biomass, because DNA has a higher nitrogen and phosphorus content than the rest of the plant cells by and large. Neofunctionalization is another thing that we talked about. So you can imagine that when just one gene is duplicated, there's the potential for the two paralogs to undergo separate evolutionary trajectories, and for neofunctionalization to occur after just a single gene duplication event. After a whole genome duplication event, then you can imagine that potential for neofunctionalization is really magnified. So whole genome duplication events, I sort of like to explain them as, not only sort of the most profound mutation, but also, imagine the organismal genome. Oh, come on, PowerPoint, don't save the auto recovery file. So imagine the organismal genome as a Swiss Army knife, and after a whole genome duplication event, you've kind of like slapped another Swiss Army knife onto that Swiss Army knife, and you're letting the tools on the new Swiss Army knife just kind of like go off on their own evolutionary trajectories. So you could end up with a lot of really, potentially interesting, useful things. I realize that I'm kind of glossing over polysomy, which is just variation in chromosome copy number. So sometimes it's the duplication or deletion of individual chromosomes without a whole genome duplication event. There's really cool stuff with polysomy. I'm going to skip over a lot of it. I just thought this paper was really cool and it didn't really fit in anywhere else. This one plant, Silene noctiflora, which is just a little kind of unassuming flower, it has variation in the number of chromosomes in its mitochondria. So within each mitochondria, they have different, circular chromosomes. And some of these chromosomes are empty, which means they don't have any genes. So in these different populations, there's different numbers of empty chromosomes. There's different numbers of chromosomes total. There's chromosomes that are unique and only found in these populations. So it's an incompletely overlapping set, even though that there are, a similar number of non-empty chromosomes. So the evolutionary dynamics of some of these genomic features are still really poorly understood. So this is just a really cool kind of sidebar study that's attempting to characterize these wacky patterns of mitochondrial genome evolution. For the rest of the talk, I'm just going to be getting into whole genome duplication events. So you probably are aware of whole genome duplication events in the context of angiosperms. This is just a bigger version of that. So this phylogeny is attempting to show all of the times that whole genome duplication events are thought to have occurred in the history of angiosperms. So you can see that in different lineages, there's differing numbers of these little green dots, and each green dot is an inferred whole genome duplication event. So you can see they're really popping up all over the place in plants. Now the usual party line is that there's not as many of these in animals. Yeah, I think actually it even says it, right here-ish. Whole genome duplication is much rarer, but has been a key event in the evolution of genome in many groups, especially plants. So this is, as of 2017, whole genome duplication events are thought to be, somewhat rare, but important for some groups of organisms. What we're learning as people look more and more for whole genome duplication events is that we can find them in other places as well. So in a pretty recent study, Jung Lee and his colleagues, including Rebecca Rundell and some other folks that I've worked with, they characterized a bunch of whole genome duplication events that they inferred genomically from insect transcriptomes. And so here you can see this whole little cool phylogeny of all the insects, and these little dots or diamonds are whole genome duplication events that they've inferred using a variety of different methods. So two of the methods that they've used have been looking at these KS plots. So what they do is they look at the evolutionary distance between genes that are in the same gene family. And if it looks like there's a bunch of genes that are sort of the same evolutionary distance apart from each other, more or less, that's consistent with the idea that the whole genome may have duplicated. Whereas if there's just kind of an even smear of evolutionary distance among all of the genes and all the different gene families, that's consistent with just sort of a steady birth-death process of, every once in a while a gene is duplicated or lost, but it's not done in tandem as a whole genome. They also inferred these whole genome duplication events using a tree-based method that I'm going to kind of gloss over it for now just because it's very, very interesting, but I want to get to this mollusk stuff. So I want to talk about this mollusk stuff in part because it's kind of an unsolved mystery. And I think that's kind of an important thing about evolutionary biology to convey to you all, is that there's stuff in your textbook that's kind of like, it's all tied up kind of neat and it's solved. But there's also a lot of open problems in biology that we don't totally know the answer to. So I'm going to share some research that I've participated in a little bit to sort of give you a sense for how we go about thinking about interpreting different types of evolutionary data. So just to sort of come back to polyploidization events, paleopolyploidy is just like an old whole genome duplication event, so an ancient whole genome duplication event, paleopolyploidy, kind of fun to say. In some cases, plant clades that have undergone a whole genome duplication event are more diverse than their sister clade. That's not 100% true. Teleost fishes have undergone whole genome duplication events. And in general, the whole sort of duplicating your Swiss Army knife logic applies to some of these, but there's not always 100%, hard and fast rules about, organisms that have undergone a whole genome duplication event are always more diverse than their sister groups. As I said before, you can, after whole genome duplication events, you can have neofunctionalization. Subfunctionalization, again, is just the two paralogous copies of the genes doing just like a subset of their former function. And I'm looking at this in the context of the mollusks. So this research is coming out of a time when Lee and Mike Barker and their group was working on a variety of different plant systems and then insects. And I came to work with them a little bit and said, hey, like we should check out the mollusk situation a little bit, see what's going on. So just for those of you who haven't taken invertebrate zoology, mollusks are actually like pretty diverse as an animal phylum. There's a lot of them. If you're an entomologist, you're not impressed by the number of mollusk species in the world. But if you study pretty much anything else, you should be fairly impressed. So there's more gastropod species, which are snails, slugs, limpets, than there are vertebrate species, for example. There's a few different hypotheses on how the different major classes of mollusks are related. This is one of them. It's probably going to hold up more or less. Some of these relationships might switch around a little bit. And they're really, really old. So you might not be able to see this from your view on YouTube, but the Cambrian explosion is here. And it seems as if some of the mollusk classes had already diverged potentially before the Cambrian explosion or around the time of the Cambrian explosion. Because very, very early on in the Cambrian, there are things that are recognizably a clam. And if you sort of maintain this idea for the way that branching happened in this phylogeny, if there was already a clam in the Cambrian, or a Clambrian clam, then the scaphopods may have diverged earlier, cephalopods may have diverged earlier. These limpets and the sort of shellless mollusks, this group, may have diverged even earlier than that from the bivalves. So it's definitely a very ancient group of organisms, and they've had a lot of evolutionary time to radiate. Snails, bivalves, other stuff. Who cares? And there's also some pretty famous examples of recent polyploidization events that have happened, or relatively recent known polyploids. So we've actually talked about this little puppy before, the Potamopercus antipodarum snail, which is the New Zealand mud snail. They're the ones that exist in a lake as partially asexually reproducing snails that are just females, parthenogenic, and partially sexually reproducing snails. What I didn't tell you before is that the parthenogenic snails are triploid. They're actually a different ploidy level than the sexually reproductive snails. So not only is there examples of sex and asex within pretty close, geographic distance of each other within sort of one population, there's also separate ploidy levels in this population. Similarly to Seagrave's diagram of sort of the small plant, big plant polyploidization event in plants, there's also some recent polyploidization events in some commercially important shellfish. So like this, Crassostria, it's a oyster. Duh. I gave this talk a few years ago and I called it a scallop, and my advisor really made fun of me for that. So it's an oyster, I swear to God. But yeah, Crassostria gigas is found sometimes as polyploid individuals, and they're bigger. They're just meatier. They have more oyster tissue for you to sink your teeth into or whatever. So they're commercially desirable. So there are recent examples of polyploidization events, or there are organisms that we know exist as polyploid individuals. So it's plausible that polyploid events play a role in the deeper evolution of these organisms. Now, another way of inferring when there were polyploidization events in the ancient history of organisms is just by counting chromosomes, right? I haven't really talked about that because it's easy to get enamored in these kind of bioinformatic approaches that just use like sequence data and KS plots like this, or this kind of gene tree based approach. But there was actually a paper that came out in 2011 that just looked at the number of chromosomes in different mollusks throughout the mollusk phylogeny, and tried to estimate when there may have been whole genome duplication events based on the distribution of chromosome counts throughout the phylogeny. So in some parts of the family tree of mollusks, there are higher numbers of chromosomes that are normal. And in some parts, there are lower numbers of chromosomes. And if the higher numbers are kind of close to twice the number of chromosomes in other parts of the tree, then that's consistent with the idea that there was a polyploidization event in that part of the phylogeny. So that's really interesting. It doesn't always work for ancient whole genome duplication events because there seems to be an evolutionary tendency for sort of re-diploidization. So chromosomes will kind of fuse back together for some reason in evolutionary time. And often after tens of millions of years, there will no longer be twice the number of chromosomes in what was formerly like a tetraploid organism. So chromosome counting can sometimes underestimate the number of paleopolyploidization events in different groups of organisms, but it's unlikely to overestimate the number of paleopolyploidization events in organisms. So this paper came out that was pretty cool, and they said, well, there's probably three. There's probably a paleopolyploidization event in the group of snails that includes land snails and some of their relatives, probably a paleopolyploidization event in some of the cephalopods, maybe not octopods, but probably some of these squiddy things, and then also potentially a paleopolyploidization event in a certain family or a certain large clade of marine snails, mostly marine snails. And so what I went in and did was tried to look for examples of transcriptomes or sequence data of just RNA from organisms that are mollusks, and see if there's evidence that's consistent with paleopolyploidization events in the same way that Lee did for the insects. So in general, kind of looking at these KS plots, if you imagine every gene in a genome undergoing paralogization and then undergoing neutral evolution, those paralogs are kind of neutrally evolving sort of to be more and more evolutionary distant from each other. If that's happening in a more or less clock-like way, that should result in a bunch of paralogs that are about the same evolutionary distance apart, if you look at just like the pairwise evolutionary distance. So we should see a peak in these KS plots. And that is actually what we see in some cases. So in general, what you're kind of potentially looking at for is, maybe there's a bunch of KS values that are really, really low, and there's kind of a long tail of KS values that are higher, but then there's this funny peak here. And if there were to be no whole genome duplication event, you'd see just more of a gradual slope. You wouldn't see this little peak here. So I'm going to skip over that. Hypsogastropoda, super duper evolutionary diverse. So one of the cool things about the Neogastropoda, that is the marine family of snails that we infer a paleopolyploidization event, that was similar to the one that was inferred from the chromosome counting approach. One of the cool things about this group of snails is that they're super duper evolutionarily successful. They're really ecologically diverse. There's a lot of species. They live all over the place. They're really trophically diverse. They do a lot of different things. They include a lot of predatory snails as well as some of the biggest herbivorous marine snails. And what you see is in this group, you see there's no peak, maybe something super duper recent there, but no peak, but then a peak here, a peak here, a peak here, and then maybe something here. We're not sure. So if you zoom in on the tree, what this sort of is showing is that these snails don't seem to have any transcriptomic evidence of a whole genome duplication event. And they fall outside of the Hypsogastropoda. So this Ampullariidae and this Ampullariid that was the top snail, and then the Cerathoidea, that's this lower snail. They both don't seem to have evidence of ancient whole genome duplication event. But these snails that are actually members of the Hypsogastropoda do. They each have a little peak in these KS plots. And the fact that the peak is in the same place in terms of what the KS value is consistent with the idea that this snail underwent a whole genome duplication event around the same time that this snail did, if evolution is happening in a more or less clock-like fashion. And so the cool thing about these snails is that they are a member of this Hypsogastropoda and siphonate Hypsogastropoda clade. So they're part of the same clade that excludes the snails that don't seem to show evidence of an ancient whole genome duplication event. And so similarly, these other members, the muricids and the cone snails, they also seem to show evidence of at least one ancient whole genome duplication event. Now the coleoid cephalopods, the squids and octopods, they also seem, according to some of these KS plots that we generated, they also seem to have evidence for an ancient whole genome duplication event. This is, I guess I should show. This is kind of cool. So these figures down here are showing the inferred diversity dynamics of different groups of organisms. So these are different groups of fishes, but this purple one is the inferred diversity dynamics of cephalopods. And so what we see here is that it does look like octopods, so Octopus bimaculotes, looks like it has a peak, and Sepiella japonica, one of the squids, looks like it has a peak, but potentially at different places, but maybe the same one. And but these look like they're more recent, and so based on the KS values, we can sort of make inferences about maybe approximately when they would have occurred. And so based on these lower KS values, it's possible that these are actually separate events that took place after these two lineages diverged, because they've been divergent for a pretty long time. The octopods and the decapods, they haven't shared a common ancestor for more than 250 million years. So since before the dinosaurs were around, really. So it looks like a lot of this transcriptomic evidence is kind of concordant with the chromosome counting evidence. The largest and most ecologically diverse clade of marine snails seems to share a whole genome duplication event, which would sort of, it's consistent with that narrative that whole genome duplication events could be good for the long-term ecological diversification of a group. Cephalopods may have undergone 2 separate whole genome duplication events. But people who sequenced the octopus genome used another line of evidence to say that they're almost positive there was no whole genome duplication event in octopods. So if there was a whole genome duplication event in cephalopods rather, so if there was a whole genome duplication event in cephalopods, we would expect it not only to result in a change in chromosome number, not only to result in that sort of hump-shaped pattern of pairwise distances between paralogs or between genes in the same gene family, but you'd also expect it to result in big regions of the genome that have the same order of genes. And so what this group was looking for was synteny. So you would expect that there would be big parts of these chromosomes, like they're trying to show you with these Drosophila, where the gene order is pretty much the same, or it's like inverted. So you wouldn't expect genes in different parts of the chromosomes to just be completely reshuffled with respect to each other. You'd expect there to be some places within the genome of one organism where the genes were in kind of the same order, because that would be consistent with this sort of whole genome duplication event. So the sort of conclusion here is that we have two lines of evidence that are independent of each other. We have chromosome counts, and we have this transcriptomic dupe pipe approach with the KS plots that suggest that there was at least one ancient whole genome duplication event in the cephalopods, if not two. But then these people who very carefully sequenced a whole genome of an octopus say that there's no way. So the octopod whole genome duplication event is really kind of up in the air right now, and it's just kind of an interesting part of doing science, is that sometimes you find a result that seems to be inconsistent with what other people are finding, and it's not immediately clear what the actual truth is. So I'm gonna cut it off there, post this on the web, and talk to y'all soon. All right, be safe out there, y'all.